Okay, and I will pass you over to Angaraj for the introduction. Thank you
Sure.
very much.
I'm very glad to introduce Dr. Michael Iacovos. Michael, I know Iacovos for, as I mentioned, quite I don't know how many years now. Then we would probably look very old. Iacovos earned his PhD in molecular genetics at the University of Toronto. And after that, he moved to Swiss Institute of Cancer Research, EPFL in Switzerland. That's where we met. Both of us worked under Douglas Hanahan, who is, for those of you who don't know, he's very well known. He wrote pretty much the Bible for cancer, I can say, probably, and continuing to write. So we were privileged to work with them. During the time, he was working on microRNAs and cellular plasticity in neuroendocrine tumors. I mean, finishing with the dog, he moved to Sunnybrook Research Institute as assistant professor, where he's working. It's at the University of Toronto. He's very popular in neuroendocrine research, as most of us, including Poe, would acknowledge, and he's doing fabulous basic and translation research, including developing preclinical models and stuff. So over to you, Echo. Was looking forward to your talk.
Thanks, Anguresh. Thanks for the introduction. Anguresh has been a good friend for, I think it's 10 years, Anguresh now, so maybe we should just go celebrate. But I learned a lot from Anguresh, especially for neuroendocrine tumors. I remember uh, he taught me so much at the beginning, so it was really fantastic to work with Anguresh when we overlapped. So what I'm going to do today, everything it's unpublished, but I will talk a little bit about two models we use here in Canada. One is organoids, and if we have time, we talk about the PDX-ovos as well, some updates. And I will show you what we can do with these models and some new therapies we think that might be good for neuroendocrine tumors. I'm also a little bit under the weather, so I hope my voice will not let me down. So let's see if I can get this going. Okay, cool. So everything looks good on your side? Okay, so I will talk about some preclinical models and some new therapies, the BH3 mimetics, and how we think we can combine them with standard of care for neuroendocrine tumors. So first, I will start with the Bible of cancer, as Anguresh said. The hallmarks of cancer is something that it was introduced by Dr. Douglas Hanahan many years ago with Dr. Robert Weinberg, in which they postulated that there are different hallmarks that are common in cancer, and one of them that is the interest for today, it's the ability of cancer cells to evade apoptosis, something that we think it's really important for neuroendocrine tumors. So apoptosis, if we look into the very basic pathway, it's really triggered by two effector proteins, the BAX and BAC, and the activity of these proteins now it's really regulated by two upstream families of proteins, the BH3 only proteins, which are pro-apoptotic, and these BCL2 proteins, which are anti-apoptotic, so they inhibit one of the functions and they inhibit the activation of the effectors of cell death, BAX and BAC. So it's being you've seen this one, the evading apoptosis was suggested almost 25 years ago now. So it's been a lot of research trying to find ways to inhibit these BCL2 proteins. And the idea is that we can inhibit these BCL2 proteins, and then we'll trigger apoptosis, and that would be a way to inhibit tumor growth. So there've been different generations of drugs. The first one, it was this navitoclax, which inhibits the top three members of the BCL2 proteins. You see these are the clinical trials. Most of them, and as you will see, most trials for inhibiting apoptosis were done with malignancies that have to do with blood, like say leukemias. The problem that they found very early with navitoclax is that it has an on-target side effect, so inhibits BCL-XL, and that leads to thrombocytopenia. But for blood cancers, it seems that BCL2 is really the most important molecule. So now what they have this drug called venetoclax, which is very specific, inhibits only BCL2, and you see there is almost 500 active drugs. In fact, this one now it's already approved for different hematological malignancies. There are other specific molecules that inhibit, for example, in BCL-XL or MCL1. But for us, what's very interesting for us is this drug called pelsidoclax, which I will discuss later in my presentation. Is a prodrug. So this one now it doesn't have the toxicity that navitoclax will have. And in fact, there was a first in human study in which they had some also patients with neuroendocrine tumors, and there is an open clinical trial again with patients with neuroendocrine tumors, including pancreatic neuroendocrine tumors. Now, if we look at neuroendocrine tumors, both for small bowel and PANETs, and you see this one is a cluster of cancer cells, small bowel and pancreatic neuroendocrine tumors, you see that in fact they express very high levels of MCL1, and this will become very important in my presentation, but also BCL-XL and BCL-W, but no BCL2. I'm not going to show in my presentation today, but high-grade neuroendocrine tumors, especially NECs, they upregulate BCL2. So even though BCL2 is not important for G1, G2, what we think now it might be important for grade 3 or neuroendocrine carcinomas. So what we want to find out first is whether this apoptotic machinery is intact in neuroendocrine tumors, because you can envision that even though you go ahead and we block all of these BCL2 proteins, there are still ways that cancer cells can evade this and can evade apoptosis. So the first question was, if we block all 4 molecules, can we trigger apoptosis? Then we wanted to see which BCH3 mimetics are effective and whether we can combine them with curative therapies. And I will show one example today. So the system we use in here at Sunnybrook is that we get fresh samples from patients the same day. Within 4 to 6 hours, we get the samples, we bring them in the lab, we disassociate them, and then we put them in these 384 wet plates, which they have a hydrogel that forms this kind of a U shape. So then the cells sit there, and they, after 3 weeks, so within 1 week, they start forming these organoids. We keep them for 3 weeks. We change media once a week. We use media with defined factors, no serum. And then at 3 weeks, usually we can do imaging, as you will see. So this is one example of an organoid. I hope this will play. So you see now this a 3D rendering of pancreatic neuroendocrine organoid, and you can see some badging in one of these. As I said, we do different characterization. Here, what you see now is DAPI. You will see some Ki67, and with red is chromatin. So if you go through this organoid now, you can see some cells that are proliferating, which are these yellow coming up with Ki67. So the cells that remain alive, they maintain very low proliferation. This was one with the high Ki67. In some organoids, we only see a few cells with positive Ki67, which is what you expect for these very low proliferating tumors. At the end, we can also do clearing, and we can do staining for different markers. Here again is chromogranin and SSTR2, as you expect. And here again is this expression of SSTR2 throughout the organoid. So again, you see this very nice membrane SSTR2 expression, as some like punctuated, as you would expect for the receptor inside the cells. So we use different techniques to validate that we have the right organoids and they maintain the characteristics you expect for neuroendocrine tumors. And then to monitor the apoptosis, we use different markers. So you see here with left, this is just a marker for a DNA. In the first panel, you see this is a chromolife marker. which was developed at Sunnybrook. It gives you the activity of mitochondria. So expect if a healthy organoid will have bright yellow. This for ER stress as red. And then annexin 5 is a marker for apoptosis. So this is a very healthy organoid. You see very little apoptosis on one of the sites here. But now if you do drug treatment, you say that the DNA is leaked, seems that this is breaking down in the nuclei. You have no mitochondria activity, and now you have very high annexin 5. And then here you see some signs of the ER stress. But for us, mainly what we use is the mitochondria activity and apoptosis for the BH3 mimetics. Then we developed some algorithms to be able to now measure the different apoptosis we have. So for example, here you see in this one, you have two different drugs, drug A and drug B. And this scale shows you the fraction of cells that are under apoptosis. So you see here on the bottom, when you don't have enough of the drugs, these organoids are very healthy. And now when you start combining drug A and B, you see the fraction of dead cells in this one here is 100%. And then that will show you, we use this kind of plots as well throughout my presentation. So you see again here, drug A, different combinations of drug B, you have very little apoptosis. But if you go, for example, here, you start getting apoptosis with drug A alone. And when you start adding drug B, you have increase of apoptosis. So as I said, the first question we want to ask is, if we inhibit now all of these BCL2 proteins using navitoclax and this MCL1 inhibitor, do we get apoptosis in neuroendocrine tumors? And again, this is an example of a healthy organoid treated on this untreated with control. This treatment only with navitoclax. This is treatment now only with the BCL1 inhibitor. So you see when we treat either with one of the two, we rarely get apoptosis. But now when you do the combination of the two, you start getting a massive apoptosis. And again, this one is a single treatment, and we monitor apoptosis 72 hours after treatment. So you can imagine if we do sequential treatments, probably this organoid will completely fall apart. So we've done this now with many organoids from small bowel and pancreatic tumors. Here I'm showing the small bowel or small intestine nets. You see again, we have primary and metastasis, both grade 1 and grade 2. And, in the general conclusion is that when we start combining navitoclax with the MCL1 inhibitor, which you see here, the different parts. So yellow, it's no MCL1. Here you're starting different concentration of MCL1. We see a synergistic apoptosis. We do see some cells that do escape. So you see here, for example, these two patients here, it seems that they're resistant to apoptosis. So what I would say is maybe like 90% of patients will respond to this combination therapy of navitoclax and MCL1. And again, there was no difference between primary metastasis, G1, G2, or male and female. And we've done this now with almost 20 patients. Now, because we have these drugs that are specific for different BCL proteins, like the navitoclax inhibit only BCL2, or this one inhibits only BCL-XL, we start trying to figure out which one of them is the most important, and do we really have to inhibit all three of them? So again, when you do combination, when you don't have any treatment, you see these organoids are very healthy. But now when you start doing combination, again, on the right side is navitoclax with MCL1 inhibitor, I get massive apoptosis. BCL-XL, it's important, but it seems that is inhibiting only BCL-XL, you don't get the same response. And BCL2, as you expect, because it's not expressed in G1 and G2, you don't see any apoptosis. So I think what we believe right now is that, using the BCL-XL alone or any product that inhibits only BCL-XL, it will not be enough for neuroendocrine tumors. So in fact, what we have to do for neuroendocrine tumors is probably inhibit all four members of this family. So from so far what I've told you, then if we inhibit all four members with navitoclax and MCL1 inhibitor, that will lead to apoptosis. MCL1 seems to be critical and very important. So if you don't inhibit MCL1, you don't get an apoptosis, or very rarely you get apoptosis. The problem is, again, navitoclax is toxic, and MCL1 inhibitors, they have two problems. They are not in the clinic yet, but also the ones that are currently in the clinic, we know they have high cardiotoxicity. So probably won't be able to use MCL1 inhibitors unless we have prodrugs in the clinic. So for us, we want to understand whether we can use pegcetaclax, which is a prodrug, instead of navitoclax, and whether we can downregulate MCL1 using basically some of the current therapies. So a little bit about pegcetaclax. So pegcetaclax is a prodrug. It's as I said, it's now in some clinical trials. So the advantage of pegcetaclax, it cannot enter the cells unless it's cleaved by some esterases that are specifically expressed in solid cancers. So in the circulation, this will not go into platelets. We will not enter any of the cells in the circulation, but only in cancer cells. Now this domain here, it's removed, and then you get this active drug that has the ability now to enter the cells and binds to BCL2 and BCL-XL, probably also BCL-W, and then trigger apoptosis. And this is the paper in which this one was first described. So we did the same as we've done before. Again, pegcetaclax, at least based on the first paper, inhibits BCL2 and BCL-XL, but based on the clinical trials, they claim now that it inhibits BCL-W as well, and I think that's the case because as we show in neuroendocrine tumors, if you inhibit these two only, you don't get enough apoptosis. So again, you do combination of pegcetaclax and MCL1 inhibition. Again, you see now that you get this kind of synergistic effect. So, and again, here we use the prodrug. So we think that if you use the active drug, or if you have more time for conversion of the prodrug to active drug, you're going to get even more robust results. We've done this now, this is in progress. We've done it with different patients, again, primary metastases, both male and female. And we see very similar results of what we saw with navitoclax. Again, when you have this combination of pegcetaclax and MCL1, you start seeing this synergistic effect. We are repeating this now with the active drug, which we think that we're going to see even more apoptosis. So I guess the second model, or second conclusion, is that we can use pegcetaclax now instead of navitoclax for neuroendocrine tumors, and I think this is very good news. The question is now, how are we going to inhibit MCL1? Because again, this drug is nowhere close to the clinic, and the current form of drugs are probably going to have cardiotoxicity. So it's been shown, in fact, that when you cause stress in cancer cells with different treatments, for example, chemotherapy, MCL1 is downregulated. So in a lot of cancers now, what they show, for example, is small cell lung cancer, that if you use pegcetaclax with chemotherapy, then you get this kind of combination effect and you get massive apoptosis. For neuroendocrine tumors, at least low grade, we don't use chemotherapy usually in the clinic. So we start looking at different standard of cares we use in the clinic. And one of them that we tested, it was Lutathera. So Lutathera, we know it binds to SSTR2. Now, at least in some centers, there are clinical trials looking whether we can move Lutathera as first line of therapy for neuroendocrine tumors. So really the question was, we went back to navitoclax. If we use navitoclax with Lutathera, are we going to see any synergistic effect? So again, here is the control, no cell death. When you use navitoclax, this is one of the very few patients where we see some response. With Lutathera, you don't see any cell death, and you don't expect to see cell death within 48 hours unless you use very high levels of Lutathera. But here again, this one within 48 hours, when you do combination, really you see that 100% of the cells in the organoid basically will die. Again, these are some representative data. It's two different patients, the primary tumor. So this is the only one of the few examples where we see just navitoclax alone in the high concentration having some activity. You see now in the liver meta from the same patient, there is no activity. But now when you do a combination of navitoclax with Lutathera, you start getting this synergistic effect. And this is a different patient. Again, from this patient, we have 3 different liver metastases. The good news is that the combination, it seems like works very well. But again, it seems that there is heterogeneity within neuroendocrine tumors. But maybe this is a treatment where we can overcome some of this heterogeneity and get the same response. So this is where we are right now. We believe that we can replace navitoclax with pelcitoclax. We can use maybe radioligand therapy to downregulate MCL1, and we're doing some molecular experiments, Western blots, to show that. And what we're examining right now is whether, in fact, using pelcitoclax with radioligand therapy will lead to apoptosis for neuroendocrine tumors. Okay, so I'm gonna switch to the second part of my presentation, which is I'm just gonna give you a bit of update of what we're doing with PDX-Ovos. We're not at the same point as the organoids, and the way I'm thinking about how we can use these 2 platforms in combination, at least my thinking is that if you do let's say a medium throughput screening with organoids, and then you decide that you have 1 or 2 effective therapies, maybe PDX-Ovos will be another model to verify these any combination therapies you think might be important. So for this one, we started with different neuroendocrine cells, not just only PANET. We also got some nice some new cell lines from Dr. Yer for SI-NEC. And the way it works here is that we receive eggs, which we crack them, we put them in these dishes, and then you incubate them for 4 days. So you wait for this coral endocrine membrane to become vascularized, and then at 8th day, you can either inject cancer cells or you can put tissue fragments from patients. And then you can, starting from cancer cells, we tested different cell lines, both for PANETs and SI-NETs, and we've seen that neuroendocrine cells have the ability to grow in this CAM model. So it seems that the CAM model was permissive for neuroendocrine tumors and neuroendocrine cells, something we didn't know at the beginning. So these are some bright field images. So what you see here is the different nodules or tumors growing from these cells. We use focus ultrasound, which we can use now to measure the size of these tumors in the CAM assays. Then we went back to establish some of the immunostainings we would like to use for these models, and that was not easy because for a lot of antibodies, we saw that the CAM will give us, you're just going to get this fluorescent signal that was not specific, but we start now optimizing a lot of antibodies we use in the field. For example, this one is from a bone 1 organoid, so you see it has the right histology. What you see here, in fact, these are blood cells from the CAM model, from the chicken embryo. The blood cells in the avian model, they're nucleated. So, and that's very nice for us. You can visualize now the blood cells in the blood circulation. This again is a bone 1 high proliferative cell line. You see very high Cas7, maintains the chromogranin, and very low apoptosis. We did the same for SSTR2. Again, the NT18LM, it's known to have very low SSTR2 expression, and that's what we see in the CAM model. The QGP1, it doesn't have an SSTR2 according to the literature, so that's the same. And these are the 2 very nice cell lines, again, from Dr. Yer. Thank you so much. This one is being published in the literature. It has very low, or is basically negative for SSTR2, so we Sometimes we see a few cells that express SSTR2, but this one now it's very high for expression of SSTR2. So I think this is the same thing with agrees with the patients and also the models that Dr. Yen has published. So it seems that when we inject cells, they do maintain their characteristics, and then we moved on to start doing some samples from patients, and now here what you see in the top is different samples from grade 2 and grade 3. Patients also did from grade 1. Again, on the top panel here, you see these bright field images. These are the tissue fragments or tumors from the patients. Again, with focus ultrasound, we can measure the tumor volume. And then we also take advantage of the fact that red blood cells in the avian model are nucleated. So we can use this now as a phase contrast with the focus ultrasound, and we can actually visualize now the blood circulation inside these tissue fragments. And again, using this one, you can measure both the volume and also the vascularity, and we can discuss whether the volume is very important for neuroendocrine tumors. So here is, hopefully this will play. Okay, good. Okay, so what you see here now as yellow and these like movements, so these are actually the red blood cells from the avian model, and this one probably it's a vessel that goes on the same plane as the screen. That's why you see this circulation here. And then in the second one, again, you see some vessels and some circulation, blood circulation in these fragments. So we've seen this in almost all of these PDX-OVA organoids, PDX-OVA models that we established. We think that's kind of really we can do injection into the blood circulation of this model, and can now do drug delivery in these tissue fragments, which are intact tissue fragments from patients. So I think we've done this from more than 10 patients now, and each patient usually will have 2 or 3 different tissues, either from primary or lymph node or liver metastases. So we established maybe around 150 PDX-OVAs so far. The take rate is around 80%, and really it doesn't change. It's whether it's a patient was a male or female, the grade, or whether it's primary lymph node or liver metastasis. So it seems that it's a very reproducible model, at least for engraftment of these tissue fragments from patients. Now we start doing some characterization of the histology. So on top, what you see is a PDX-OVA, and below is a section from a human patient, corresponding human patient. So the first thing you see is that they do maintain these NETs clusters, which are very characteristics for neuroendocrine tumors. So this is really good. You see both here in these 2 samples. One is primary tumor and liver metastasis is the same. Now you see that the human sample has way more cancer cells, but for us now it's very difficult to say whether it was 2 different fragments that we got from the same patient or whether there is something else that is happening. So that's going to be very difficult to resolve. But I think at least what we see is that we maintain these very nice structures. And what you see here, in fact, is these are red blood cells from the avian model. So you see again, the circulation from the avian model is very close to these NETs for cancer cells. We did the same thing as the organoids, right? We did Ki-67 staining. So these are 2 tumors, like this. The top one is G2, but just let's focus on the middle and the one below. So this is a patient, from the same patient, OCC9. So this is a primary tumor. They know that the primary tumor from this patient was grade 1. So you see very low proliferation, just 1 cell proliferating. And this was a liver metastasis from the same patient. And in this patient, the tumor switched from grade 1 to grade 3. And again, now in the metastasis, again, you see now much higher proliferation compared to the primary tumor. And I think that's very important, again, showing that this heterogeneity that we see in neuroendocrine tumors in patients, we can recapitulate that when you use PDX-OVA models. We looked at at CLIF-Caspase-3 for apoptosis, and we see very low CLIF-Caspase-3. So again, that kind of agrees that this is a good model for this tissue engraftment and establishing of PDX-OVA for neuroendocrine tumors. We are very interested on radioligand therapy, and so with this staining for SSTR2. Again, here is the same patient of C9 I showed before. This is the primary tumor, which is grade 1, and this is the liver metastasis, grade 3. What you see on top, PDX-OVA, below the matched human sample, PDX-OVA, and the human sample. So what you see is that in the primary tumor, you have very high expression of SSTR2, almost homogeneous everywhere, and we keep this same, it's PDX-OVA. We do have some necrosis here, and again, this is very difficult for us to say for sure now, is this necrosis because maybe there was necrosis already in the sample, maybe because of treatment. And when you have necrosis, you see the common granular is not as nicely stained, and you don't have SSTR2, but it also might be sample processing. We're looking into this now. But again, when you see SSTR2 in these very healthy nets of islets here of neuroendocrine tumors, very high expression. Now, if you look at metastasis from this patient, again, who switched from grade 1 to grade 3, you see that in the metastasis from the patient sample, you have very few cells with SSTR2, and that is the same for the PDX-OVA. Again, showing that both in the case of Ki67, but also SSTR2, it does maintain the features you have in the patient and this heterogeneity. So I think that's promising, I would say, for this model. And I mean, my thought is that we have to understand the model and what our model can offer us, and then based on those advantages, we have to ask the right questions for each model. We start looking a little bit into the microenvironment. Again, the challenge here is that we have to find specific antibodies that, for example, differentiate the human from the microenvironment coming from the PDX, from the chicken, from the avian model. And that's not easy because, as you can imagine, not many people characterize the antibodies, whether they have any cross-reactivity. We use vimentin. So vimentin is a marker that at least is used in our pathology for mesenchymal cells in the stroma. So you see here is a human sample, and you see you have some stroma cells, but also some pericytes. They are also positive in vimentin, some of them surrounding these tumors. And now this is the PDX-OVA model. We do get some vimentin in the stroma. Again, these are some vessels, but we're looking into it. We're still not 100% sure if this is just vimentin from human samples, or if some of the avian fibroblast ones, they move into the graftment and then they upregulate vimentin. So we're still looking into this. And we're looking also whether there are any immune cells, either from the patient or whether there are some immune cells that are recruited from the avian model. All right, so I think I guess what we are certain right now is that the take grade for neuroendocrine tumors, grade 1 and grade 2, it's very high in the PDX-OVA model, and I imagine it's going to be even higher if we use grade 3 or neuroendocrine cancers. They are vascularized, that gives us the advantage now that we can do drug delivery through the blood circulation. They do remain viable. We keep the same Ki67 as the patient. And I think what is very exciting for us is that we do maintain the expression of SSTR2. And this was my last slide. I would like to acknowledge especially two people in my group. Dr. Nilakshi Kulathunga is doing most of the work in the PDX-OVA, and Zoe Wang is a PhD student in my group. She's doing most of the work with the organoids, the different collaborators. Dr. David Andrews, who help us with the organoids. Justin Kell does most of the image quantification. Dr. Hong Leong and Yan Li, they help us with the PDX-OVA. Alberto from Dr. Carbonero was here for one summer and really helped us propel the SSTR2 and Lutathera treatments. Dr. Paul here from different cell lines we got. My collaborators at Sunnybrook, Dr. Singh, Halen, and Law. and of course the funding agencies and all the patients for consenting to give us samples and use them for our clinical research. So I'm happy to take any questions and discuss further.
Thanks, Iacovos. That was a fabulous talk and very comprehensive and interesting. And need of the hour for this cancer type because not many models out there. And apparently, the resources that you're developing, I believe will, progress this field much faster than, I think, in the past it was stuck because of lack of models and stuff, which is great. Yeah. Any questions for Iacovos?
Yeah, I had a question. Terrific work. Really interesting to see that, and such a great patient number as well for your PDX-Ovos. I had, you looked at male versus female, and also looking at grade kind of take rates. Do you have like a functional tumor cohort group in your kind of patient set, and have you considered looking at the differences in take rate between the two? Or if they're maintaining any functionality after they've been basically reimplanted, because I think that'd be really interesting to see in this.
I mean, that's a very good question, and we never thought of this. You are right. Yeah, I don't know if we're going to have for panels any solenomas. We just had one last week, which was grade 1. You're right, we should go back and look into this. I guess for assignments, what would be the suggestion for functionality? What can we do to prove whether they're still functional?
That's a good question. I guess it kind of depends on what exactly. I'm not sure of your numbers, your breakdown between the two groups. But if they're, and I'm not a clinician, I'm a postdoc, so I'm maybe not speaking exactly the perfect terms, but if they're serotonin producing or serotonin kind of functional tumors, what do they produce off the top of my head? You can maybe look into functionality there. But I guess it kind of depends on where exactly, what your numbers are. And yeah, I'd have to look into it a little bit more, honestly, for those ones.
I'm going to go to my great collaborator, Dr. Poyer, and you have some tracers for serotonin activity, right? Maybe we can use them. We can see whether.
Yeah. And I think so most of the, ileal neuroendocrine tumors express high levels of serotonin compared to the other neuroendocrine tumors. Not that they don't express. I think they do too, but I think in particular it's just like you detected more in the ileal net because I think, they're packaged into like these vesicles with and they co-stain really well with the, VMAT1 as well. But yeah, I think we put out like a paper on developing like for the sensor for a luciferase based sensor for serotonin. I think it's definitely hard for us to do it in mice still, but I think if we do a more reductionist model and a more simplified model, maybe the ovo model could work. And thank you again for Iacovos for showing the nice neck cell lines that form these little tumorlets in your ovo model. I was like, oh my gosh, that's so pretty.
No, it's
well, thank you so
really
much.
amazing. They grow very well. Yeah. Thanks for the suggestion, Stephen. I'll definitely follow up for the serotonin sensors. That would be really great. Thank you.
Is it the ghrelin? I don't know if I'm pronouncing the right way. I think that's probably one of the molecules that you would look for functional activity. I think Benjamin working on a publication. I think one thing. But I'm not an expert. I'm not familiar. But yeah. Any
I think it's a fantastic work. So I wonder, and what's the difference between your work and our work? We've been working, for the models for the 4 years. And we have presented our work in last year in the, I know, and Dr. Mark know our work, Dr. I know our work. So today I have one question. And I'm thinking why we need to, and spend so much time to do the genetically modify, models. and why just not to do this? So I have one question. So the model, your PDX or PDO, you just, I know, the G1, G2, and the NET, you do the PDO, right? The
Yes.
cell can just sitting there, they are alive.
Yeah.
But it's just for the organoid, sometimes, for my opinion or my experience, it's just some cell clumping. It's not like, maybe it has some leaching selection, some stromal cells is gone, or you still leave some tumor cells and clumping and looks like an organoid. But it's really hard to proliferate. It's like usual. The Ki67 very low the percentage. So I wonder, you just get the cells and play with it and treat the drugs and that's it. No more material left. Maybe that's a difference between yours and ours. That's one. A second one is for the PDX. I really, like all the picture you show, and it's beautiful picture. It's like mimic the parental tumors. But it's the same question. It's, what's the practical and or applicable in the drug treatment if you use these? It's just how many samples or you can get from clinic, just put this and to get the PDX hours and enough numbers for the drug treatment or whatever things you want to do. So maybe that's a significance in our models. I've been thinking about it. We know, once you put the genes, although it's a doxycycline inducible, we might change something. I know everybody, concerned about that. Although we confirm is in the large extent is maintained the parental in the genetic and molecular or transcript or whatever levels. But for yours, I really like it. If we can, get enough for the application. So my question is, how practical applicants for your models in the translational study in terms of, and understanding the no grade NET and the mechanisms or the translation drug screening or whatever we can think about it. So that's my question.
No. Thanks for the question. And I think, I kind of alluded this between the switch of my presentations from PDO to PDX OVO. For me, at least as a basic researcher scientist, the way I would like to think is that in each model, you have to ask the right question and then you have to move to the next model. And there is not going to be, especially in neuroendocrine tumors, there's not going to be a model we're going to be able to answer all questions. And you are right that if somebody doesn't have the samples that we get from the clinic or a biobank that runs the way it runs here at Sunnybrook, it's going to be a bit difficult to do this to the way we do it. So for the organoids, just to answer your question, for the organoids, usually we get a sample almost every week. It's a patient every week, and usually we get 1 or 2, sometimes 3 samples from the same patient. And right now we can do at least 1,000 organoids per sample. So that means we can run around maybe 5,000 organoids per month. But again, what we like to do is start, with a set of questions instead of doing high throughput. In this case, we do apoptosis. We try to show the same thing for 10, 20 patients, and then we are sure that that pathway is active. And then you move to PDX OVOs. PDX OVOs, from each patient, usually from each sample, you can do 8 to 10, maybe 12, right? Because now you cut fragments.
Yeah, I understand.
And then you have to talk to your pathologist. You have to make sure that they give you the right sample and all that. So again, now if you have a patient, you have 3 samples, you establish 30 PDX OVOs, and you want to do a treatment with 3 arms, and you want to do 10 PDX OVOs per arm. So, again, you have to figure out what you like, and you do it in the PDX OVO. And what the PDX OVO is going to show you is how the patient is going to respond. And again, you have to understand that the PDX OVO you understand maybe why the patient will not respond, right? That's probably within the time frame of PDX-OVO. Now, if you want to see whether there is a resistance, then maybe the models you are developing, because now we can go to mouse samples, to mouse models, then we can do longer treatment, right? Let's say we work something from the organoid to the PDX-OVO, but then now we want to say, okay, if we treat for longer to have resistance, maybe then we use some of the great models you are developing. So again, for me, it's like, we have to do the right question for the right model. And I think everything
maybe after our model screen some, and target drug, maybe we can confirm in your model. I think that probably combine our two models.
I would love to if yeah, I mean, we can talk after, but if you want to send us some of the models you develop and put them in the PDX-OVO and see whether they keep some of the characteristics, or that would be also great.
We do, recently we do and screened and a bunch of the FDA approved anti-cancer drugs. We do like to get like 10 around the candidate case. We're gonna, conform. Yeah. I think, combine our both work probably.
No, I think it's you have to know your model very well and really ask a question that is the right question for the model.
Okay, thank you.
Thank you.
So taking from that point.
I just want to introduce myself. I'm working. I'm the Anderson Associate Professor, and I've been working with the GI and cancer. And 4 years ago, I work with Dr. James Yao. I know probably a lot of people know James Yao, who is the pioneer in the neuroendocrine tumor with my chair. So he, in his life, he tried to establish model many times. Yeah. He liked the current model. So we're now where we got, we just got and Texas. And it's equally like our one, the funding to do the drug screening, use our model. So and we are very likely, go into this field. So I just want to stop here.
Thanks, Singh, for introducing yourself, and we're glad to meet you. Great, good question. But following up on the point both of you discussing, I just wanted to also mention that Iacovos knows that we are just on hunt for a syngeneic model, because the right question, the right model is important. We are modeling one of the metabolic stuff, and without the immune system, the effect is it's nothing, no effect there. So again, it's a pain that we are just going through and getting some help from Iacovos. Great. I have a couple of questions. So it's great that your apoptotic inhibitors are combination working, and I also noticed that it's just one microgram or less where with the synergistic effect. But do you think that the downstream signaling would be very different from one drug to the other? Because Paul Workman, who used to be a previous CEO and who developed this drug unit at ICR, and he's a very popular guy in that area. He always mentioned that even for the same target, two drugs could work differently. Here you're trying to really target multiple anti-apoptotic ones. So do you think that there will be a very different signaling events that you were targeting there?
Yeah, that's a good question, Ankuraj, and I think probably these questions have been answered in the original models, because when they publish these molecules, because they really do very sophisticated biochemical assays to show which targets they develop, because they target, because you are right, maybe the affinity is difference between the three molecules, right, between nab-deoxyribonucleic and paclitaxel. I assume that's what you are asking.
Yeah, that could be done before, but I'm also, in my experience, some of these drugs, not necessarily very big difference, but it could be context dependent and also combination dependent or whatever, right? So just was curious, yeah.
Yeah, I think that's why it's, at least with the organoids, it's very important that we screen as many as possible and we see how reproducible it is.
Right. In your OVO models, I know it's a bit tricky, but where maybe you mentioned I missed it, but did you try characterizing any metastatic events? The cells spreading into, because why I'm asking this is because our neighbor, they're doing in sarcoma, which we are kind of adopting for colorectal. And again, we could see some. So I'm just wondering if neuroendocrine tumors also do something similar.
In the same model, Ankuraj?
OVO, yeah, OVO model, yeah.
So in other, we haven't. You know what? The reason is probably I didn't think we're going to see metastasis, so we haven't looked into it. I know for other aggressive tumors, you are right. Like even breast cancer, for example, you see it, right? We haven't looked into this. We should.
And also you could probably stain for ALU sequence. There are antibodies against it because you were mentioning about this vimentin coming from. So we actually, how do we do look at the metastasis is just by PCR, ALU PCR in the other areas and see if there are, because ALU is present only in the human, so look for the human cells there. And
Really?
there is also
Okay.
an antibody as well. So that's where you could probably try to differentiate between whether the protein is from
Oh,
the.
that would be great. I will definitely follow up on Crash because we tried MHC, we tried so many antibodies, and we always see sometimes this autofluorescence, especially on the outside of the cam, and we are not. But yeah, if you have an antibody that works, that would be
We can offline. We can discuss. Yeah, we can help you with that.
absolutely. Yeah, that's a great idea. Thanks so much.
Any other questions?
Yes, I'm Renata, and I think we know each other from Toronto meeting.
How
And
you
I'm
doing?
really happy that the Terry Fox Foundation really helped with this, with those projects.
Absolutely. Yeah.
You
I
know,
know.
just technical thing, we see actually quite a lot of SSTR2 in QGP cells. So if you want to try our cell line, even we published, so our QGPs have quite a lot of SSTR2. You know, another thing, what we notice if aggressive G3 are not positive for SSTR2, they are quite often positive for UPAR. So UPAR is urokinase plasminogen receptor, and there is a peptide targeting the UPAR. And there's a couple publications, I don't remember the author, but I think he's from Europe and he's a Dutch scientist. But when you will Google UPAR targeting and neuroendocrine tumors, and I think the peptide is AI115.
Okay, great.
And I think he was even some time ago awarded by NETChatter.
Nice. Okay, yeah, I will follow up on this. Sorry, what was the cell line you mentioned?
QGP. So our QGP is positive for SSTR2, and I think you show that you could not really.
Oh, you are QGP. Yeah, okay. Yeah, okay. Okay, maybe I'll follow up. Maybe I'm not sure how easy it is to get it, but yeah, it'll be great.
Yeah, but you know, my question is, so the another cell line, NT18LM, is also because we have NT3s.
Yes.
It's also from Dr. Jörg Schroeder.
Yes.
Okay, so he has more.
Yes.
More
And
to share.
so
Okay, so
we got the NT3 now. We'll see. Very slow growing, but yeah.
Fantastic work.
Thank you so much. Thanks.
Any other burning question from anybody?
Oh, I have another question, and I just want to get our expert opinion. So we found some phenotype. It's like cell line, if you culture it in 2D, it's easily to lose the neuroendocrine markers. Once you put the same cell line back to the mice, the neuroendocrine tumor marker come back. Do you guys see this?
Yes, I can confirm. We have that experience too.
the same thing? Oh,
I
okay,
think, yeah,
okay.
because some of these cell lines, I think if when you culture them like beyond 3 months or something, and especially in 2Ds, they could lose the expression. But yeah, if you put them back in mice and then re get the tumor and get the tumor cells, and you can, and we've only tested most of the time with the SSTR2 receptor expression, but I'm sure if you test for other markers, you probably find the same.
Oh, okay, okay. So we don't know yeah, we just see this,
Yeah. It would be nice to test maybe just if you do organoids. Paul, have you done that to see if it also regains the SSTR2 from organoids? Because is it the microenvironment or is it the 3D floating structure and this paracrine signaling maybe that promotes?
Yeah, I think it's the 3D structure because I think
yeah,
it's
contact dependent.
matrix. Yes, yes.
Yeah,
I don't
I do
really know
see
the
it's like
pathway
2D is the
yet,
worst
but
and 3D in the middle, and the PDX is the best. I saw this.
Yeah, so I think I mean, I know it's painful, but I think we should try to keep these cells in suspension for most of our studies at least.
Okay.
Any other questions? Okay, if not, that was it's really great and pleasure
Thank you
to
so much.
leave it to leave.
That was
Thank
great.
you so much, Dr. Michael. That was really interesting. Thank you for giving us your time. And thank you everyone for coming. Just a quick reminder that there's 6 days left to register for the next model meeting, which is being held on the 11th and 12th of September in Oxford in the UK. So if any of you, I know some of you have already applied. So if anyone else is interested, then please do get your registrations in within the next 6 days. That'd be great. Yes. So Angie Po, yes. So thank you so much everyone for coming. And the next meeting is on the 29th of May. So hope to see you all again then. Thank you very much. Thank you. Thank you so much.
Bye.
Thanks.
Thank you.
Bye-bye.
Bye.